You know, if there's one molecular structure that just about everyone recognizes, it is good old DNA. And it is indeed an important molecule. We have it in our body. Here's mine. But seriously speaking, it is the so-called blueprint of life because it directs just about every process in the body. You know, we normally say that science marches along by small steps, but the truth is that every once in a while it takes a giant leap. And that giant leap happened in 2012 with this paper published in Science, one of the premier journals of the world. And as you can see, that headline, that title looks like some incomprehensible word salad to anyone who doesn't work in the area of biotechnology. But those who are in that area, to them, this was an absolute revelation. It changed their life, at least as far as laboratory work goes. And Emmanuel Charpentier and Jennifer Doudna, the main authors of that paper, went on to receive the 2020 Nobel Prize in Chemistry for this work. What is this work? Well, in order to tell you about this complex business in a relatively simple way, we're going to do a little bit of origami. DNA is the molecule that, as we said, is the blueprint for life. And it is made up of small segments called genes. And each of those genes codes for a protein. Proteins are not only the building blocks of our body, of our flesh, but they are also enzymes. These are biological catalysts that make all of the reactions that constitute life happen. Well, if we can inactivate a gene, then it means it can no longer produce a specific protein. And that can be very important. And that is what that paper was all about. It basically described a technique known as CRISPR-Cas9, which essentially describes molecular scissors that can cut out a piece of DNA. So let's do our little bit of origami. What the researchers learned, and they learned this from bacteria, because when bacteria are attacked by a virus, the bacteria learn to take a piece of the DNA of the virus, embed it in their own DNA, so that next time they're attacked by a virus, they can react to it. The bacteria remember it. Anyway, it was that technology that the researchers used to basically be able to cut out a gene from our DNA. And what then happens is that the DNA repairs itself, but there's a gene missing, which means that the protein for which that gene was coding is also missing. So what, you may ask? This has tremendous possibilities. For example, there are diseases that are caused by mutant genes. And if that gene can be removed, you have a possible therapy. Things like sickle cell anemia, muscular dystrophy. That's fascinating. But many, many other applications. For example, cassava, which is a tuber that feeds millions and millions and millions of people in developing countries. But if it's not properly processed, it has cyanide in it, and it can kill people. But you can inactivate the gene that codes for the production of the compound that releases cyanide, and this can be done. Other things, for example, the Japanese already have a product on the market, and this is a very interesting product. It's a tomato. And as you can see, it is a tomato that is enriched with GABA, gamma-aminobutanoic acid. They've done this by inactivating a gene that would break down this particular chemical that is naturally present in the tomato. So what? So what, you may ask? Why is this important? Because GABA is a naturally occurring compound in our body that can work as an anti-anxiety agent. And furthermore, it can lower blood pressure. So it is possible that this tomato, which is already on the market in Japan, is going to replace some medications for blood pressure. So indeed, this paper was a giant leap, and now we know how to edit DNA. And you don't have to be concerned that we're going to be choked by this. This is not genetic modification in the traditional sense. No foreign genes are introduced, as in GMOs. Only a gene is removed, which means that all we're doing is getting rid of a protein. Fascinating technology, well deserving of the Nobel Prize.